Baltimore class variations. A presentation by Professor Gershom Zajcek, Faculty of Medicine, Hebrew University of Jerusalem. Introduction. I describe the original Baltimore classifications and suggest two updates. One, classification of processes, and the other update is classification of cell dynamics. Baltimore classification is a system used to classify viruses based on their manner of messenger RNA synthesis. 7 Baltimore groups are described that take into consideration whether the viral genome is made of DNA or RNA. So here you have the original version of Baltimore classification of viruses. You have 7 groups of viruses which interact with the cell in order to get access to messenger RNA. Now, if we manipulate it a little and add here some receptor, and so you see that the story is virus infects a cell in order to get access to the messenger RNA. It's an interaction between cell and viruses. Now, if we would like to change this interaction into a process, viruses are processes interacting with a provirus. So you add here the provirus, and the whole thing becomes a virus's processes interacting with provirus. A provirus is virus genome that is integrated into DNA of the host cell. In the process version, provirus occupies the center stage of the host cell. So here you have the provirus. It is a set of cell genes with 3 functions. It stores the self of a cell, controls who may infect the cell, and controls virus production. And self is a set of provirus genes unique to cell. And I described this in the theory of virus and cell update. So what we are talking about, virus attaches to a receptor, which is an infection, and triggers the formation of the provirus, which then is responsible for the 3 functions which I just described. Now, if we make out of this unit a provirus process, so you have a virus, attaches to a receptor, interacts with or triggers the provirus, and the provirus provides the virus. So, virus infection triggers a cell process driven by provirus. 7 processes triggered by 7 virus classes are represented here. So you have here the 7 processes which are triggered by 7 groups of viruses. Now the second version which I suggest is based on cell dynamics. Cell dynamics says virus triggers and cell decides what next. And so here you have the dynamics of cell and virus, which I describe this idea. So virus triggers, attaches to a receptor, and it triggers the cell, and the cell has to decide what next. And the cell has 2 possibilities. Either the cell creates viruses, which is lysis, or the cell decides to add its information to the genome, which is lysogeny. And this information is endogenous viral element. So this is the unit of cell dynamics, and we are interested to make out of it a process. So in terms of process, you get virus triggers, attaches to a receptor, and cell decides, is it going to lysis or whether the cell is going to lysogeny. Now, if you take this idea for all the 7 groups of cells, you have viruses, triggers, and cells of their origin decides. So this is a second update which I suggest for Baltimore classification. Why all the toil and trouble? Because we are facing the Virchow's law. Virchow's law says all cells arise only from pre-existing cells. And I think that we have to add this, introduce this Virchow law in these classification tables. And this is what I try to convince you. So what the law says? The law implies that virus is created by cell, virus is a cell organelle, and virus is endogenized, virus is not alive. So all these features are now as processes. Conclusion. Virus triggers a cell type in which it was created, which is known as viral tropism. More to come. Enjoy.